Select the incorrect statements with reference to mitosis. So the first statement is splitting of centromere occurs at anaphase. Yes. All the chromosomes lie at the equator at metaphase. Yes, it's correct. Spindle fibers attach to centromere of chromosomes. No, they attach to the kinetocores of the chromosomes. Chromosomes decondense at telophase. Yes, so the correct answer would be the third one, because this doesn't occur because spindle fibers attach to the kinetocores of chromosomes. So this statement is incorrect with reference to mitosis. Let's see why the solution. So at metaphase is characterized by the chromosomes coming to lie at the equator, which is the correct statement. Spindle fibers attach to kinetocores and not the centromeres as mentioned in the statement. And chromosomes, yes, again the equator statement is correct about the chromosomes at metaphase. Next statement. So at anaphase, what is happening is the following key events, which includes the splitting of the centromeres where the chromatids separate. So here we can see how the centromeres have splitted and the chromatids have been separated in the anaphase stage. Next, in telophase, the chromosomes have reached their respective poles and they decondense. So after the anaphase, we have the telophase, which can be seen over here in the diagram. So there is decondensation of the chromosomes that has taken place. Here the chromosomes were condensed before this stage, but here after the splitting, we can see the decondensation. After they have reached their respective poles, after they have reached the poles, there is decondensation of the chromosomes that has taken place. We cannot see a condensed proper chromosomes at the telophase stage. So the third statement would be the correct answer. Next, which one of the following never occurs during a mitotic cell division? So coiling and condensation of chromosomes, yes, it does occur. Spindle fibers attached to kinetocores of chromosomes, we did see. And then movement of centrioles towards the opposite poles, yes. Pairing of homologous chromosomes, no. This occurs during meiosis. Let's see the solution. So in meiosis, what is happening is in the prophase, in meiosis 1, and during the prophase 1 stage. So first we have the leptotene, then zygotene, then pachytene, then diplotene and dikinesis. So after the leptotene, we have the zygotene. During this stage, the chromosomes start pairing together, and this process of association forms the synapses. Such paired chromosomes, these chromosomes which are paired, are nothing but the homologous chromosomes. So that is why this occurs during meiosis and not mitosis as mentioned in the statement. Let's see the diagram. So this is the diagram of meiosis where we can see the pairing of the homologous chromosomes. These chromosomes, this one is a pair of a homologous chromosome and this is a pair of a homologous chromosomes, which is happening during meiosis 1, here also in prophase. So in prophase, as we can see that these are the homologous pair of chromosomes which have paired with each other, which is happening in meiosis. While in mitosis, what happens? There is no pairing of homologous chromosomes. So we can see that these chromosomes are being placed at the equator where there are no pairs of homologous chromosomes that we can see. So that is not happening during mitosis. So the correct answer would be the fourth one, since they are asking regarding the mitotic cell division and not meiotic cell division. Then the next would be next question, the appearance of recombination nodules on homologous chromosomes during meiosis characterizes what? Terminalization, synaptonymal complex, bivalent, or sites at which crossing over occurs. So the answer would be the fourth one. Let's see the solutions for this. So in meiosis 1, during the prophase 1, after the leptotene, we have zygotene. During the zygotene, we can see the pairing of the homologous chromosomes and the formation of the synapses. And then this stage indicates the chromosome synapses and is accompanied by the formation of a complex structure which is also known as the synaptonymal complex. The complex formed by the pair of the synapsed homologous chromosomes is nothing but a bivalent or a tetrad. These are more clearly visible at the next stage. The first two stages of prophase 1 are the short-lived ones, and the next stage, that is the patchy team. During this stage, the 4 chromatids of each bivalent chromosomes are clearly visible, clearly appear as a tetrad. This stage is characterized by appearance of a recombination nodules. So this is characterized, so here we can see the appearance of the recombination nodules. And what are these? These are the sites of what? Where at which the crossing over occurs between the non-sister chromatids of the homologous chromosomes. So therefore the answer would be the fourth one. Next, regarding meiosis, which of the statements is incorrect? So 4 haploid cells formed at the end of the meiosis. Yes, correct. The 2 stages in meiosis are meiosis 1 and 2. Yes. DNA replication occurs in the S phase of meiosis 2. No. Pairing of homologous chromosomes and recombination occurs in meiosis 1. Yes, as you've seen in the previous questions. The explanation for this, I don't think so I have to give it. We'll see why the other statements are right or wrong. So here is the explanation, where the meiosis involves 2 sequential cycles, 1 and 2, understood, but only a single cycle of DNA replication. So this DNA replication happens in which stage? It happens during the interphase stage. So after the interphase, we have the M phase, that is mitosis or meiosis, which happens. So here they are saying that we have only single cycle of DNA replication which is happening during the S phase. So in the S phase we can see the G1, S and the G2, and then followed by the mitosis or meiosis. But here we are speaking only about the meiosis. So meiosis 1, before meiosis 1 we have the replication of these chromosomes which has already taken place in the S phase. So we are having this S phase, and then the cell is entering the meiosis 1, we can say this. But since they have said only a single cycle of DNA replication, and the question has mentioned about S phase taking place in the meiosis 2. So let's see. So the next statement, 4 haploid cells are formed at the end of the meiosis 2. This is also correct, and homologous chromosomes are being paired in meiosis. Now let's see what happens before meiosis 2. So meiosis 2 is initiated immediately after the cytokinesis. So if in interphase I have the S phase and then I have the meiosis 1, then there would be cytokinesis where there would be separate 2 cells. Now this has to again enter meiosis 2. So here there is no S phase, that is meiosis 2. So here there is no S phase that can be seen. Directly enters the meiosis 2 cycle for completing the division. So this statement would be wrong for the S phase taking place in meiosis 2. So coming back to the statements, so DNA replication occurring in S phase of meiosis 2 is wrong one. So this would be the correct answer. Then next question, which of the following statements with respect to endoplasmic reticulum, they are asking about endoplasmic reticulum, is incorrect. So here the first statement mentions SER, that is a smooth endoplasmic reticulum is a site of lipid synthesis. Yes, it's correct. RER has ribosomes attached to ER. Yes, it's correct. And SER is devoid of ribosomes. Yes, it's correct. In prokaryotes only RER is present. No, it's wrong. Why? Let's see. So this would be the correct answer. Let's see the explanation for this. In eukaryotic cells, they have membrane bound structures or organelles, like apart from the nucleus there is endoplasmic reticulum, there is Golgi complex, lysosomes, mitochondria, microbodies, vacuoles. But this they are talking about the eukaryotic cells which has the membrane bound organelles. But in prokaryotic cells they lack the membrane bound organelles, and ribosomes are nothing but the non-membrane bound organelles which are present in all the cells, in both eukaryotic as well as the prokaryotic cells. So that is why the presence of RER cannot be seen in prokaryotic cells as mentioned in the statement. Let's see the more explanation. So the endoplasmic reticulum bearing ribosomes is known as the rough RER, and the one in the absence of ribosomes is known as the SER. RER is involved in a protein synthesis. SER is involved in synthesis of lipids. So all these statements are correct. Let's see the diagram. So here in the diagram, smooth endoplasmic reticulum shows the absence of ribosomes, while the RER shows the presence of the ribosomes. So therefore the correct answer would be the fourth one. In prokaryotes only RER are present, which is the wrong statement. Next question, these are the match the column for types of chromosomes and the explanations. So for the metacentric chromosomes, we can have the centromere will be in the middle, forming two equal arms. In metacentric, we have two equal arms of the chromosomes, while in submetacentric, we have the centromere situated slightly away from the middle. That's why it's known as submetacentric. In acrocentric, it would be slightly situated close to the one of the end, where we have one short arm and one long arm because of this kind of position of the centromere. And in telocentric we would have the centromere located at the terminal end. So according to this, the answer would be A 3, B 1, C 4 and D 2. So the answer would be the second one. Let's see the explanation for this. So the metacentric chromosomes have centromere in the middle, forming two equal arms, while the submetacentric would have slightly away from the middle, resulting in one short and one long. And when the acrosome would have centromere situated close to one of the ends, forming one short and one long arm, while the telocentric would have the centromere position at the terminal end. So in telocentric, we would have it like this. The centromere is at the end of the chromosomes. So this would be the telocentric chromosomes. So these are the explanations. Thank you.